Hello friends, I am Dr. Dinesh Kumar Mishra, Professor of Pharmaceutics, College of Pharmacy, IPS Academy, Indore. I am back with another episode on packaging. In this episode, we are going to talk about packaging concept and its components, specifications and evaluation methods, stability aspects of packaging and packaging equipment. So friends, let's begin our today's episode. Concept of packaging. Before going in much depth of this topic, we are going to try to learn what is the basic concept of packaging. What is packaging? It's a process by which the pharmaceuticals are properly packed to retain their therapeutic efficacy since the time of packaging till consumption. Packaging may be defined as an art and science that involves articles preparation for transport, storage, display and use. Now what are the objectives of packaging? Basic objectives of packaging involves protection, presentation, identification, information, convenience, compliance and containment. Now packaging safeguards against exterior environmental circumstances like temperature, humidity, gases, light, pressure, etc., mechanical shock and misuse. Presentation helps in image building of a product. It guides in product identification and various desired information that is displayed according to the product level requirement. Convenience is also provided for use as well as for product administration, for example, eye drops, ointment tubes, metered dose inhaler. Packaging is considered as an integral part of any pharmaceutical industry. It is very vital in pharmaceutical industry that the selected package must maintain the integrity of the product. The package selection commences from the product, physical and chemical characteristic determination, need of protection and market requirements. The selected materials for packaging must have following properties. It must protect the product from environmental surroundings. It should have no interaction with the product. It must be non-toxic. It should be FDA approved and it must comply with the regulatory requirements. It should have adaptability and it should be economic also. Now what are the packaging materials that can be used? It includes glass, metals, rubbers, plastics, fibrous materials, films, foils and laminates. Now we have to talk about what are the factors that may affect the selection of packaging material. The factor may be the product or pack contents, product application, stability of content and necessity of protection against any environmental conditions, reactivity of the content with relevant to the packaging material, pack acceptability to the customer or user, the process of packaging, issues related to regulatory, legal and quality. Now we must talk about the product packaging design. Now it's a key element of validation. The package must be designed to withstand the rigors of sterilization, transportation and storage. So these conditions must be tested for each package design, emphasizing various packaging material combinations. Packaging design must be as per the needs of the product, manufacturing and the distribution system. It should include proprietary name, dose, approved name of API, uses, instruction, prescription drugs, straightforward design, OTC drugs, distinctive design. Now we will talk about what are various types of packaging. There are three types of packaging. First one is primary packaging, secondary packaging and tertiary packaging. Now what is primary packaging? Primary packaging, that is in direct contact with the product. It maintains product quality. Now what are the examples of primary packaging? It includes glass, plastic, metals, rubbers. Second one is secondary packaging. It contains product and primary pack. It offers presentation and protection. The examples are paper, cardboard. Now next one is tertiary packaging. Tertiary packaging, this is offering transport, shipping, warehouse storage, bulk handling and the examples are boxes, shipping containers and drums. Components of packaging. Package consists of following components. First one is container, in which any product or medicine is placed and enclosed. It is usually in direct contact with the drug. Second component is closer. It is the component that tightly packs the container to eliminate oxygen, carbon dioxide, moisture and microorganisms and also prevents the loss of water and other volatile substances from the product. Third one is carton and outer covering. It is the outer casing that provides secondary protection against mechanical and other environmental hazards. It also serves for the demonstration of written information. The cartons are usually made up of cardboard, molded wooden pulp and expanded polystyrene. Another component is box. In this component, multiples of products can be packed. It provides primary resistance against external hazards and has shock absorbing features. The boxes are generally made up of thick cardboard and wood. Now we are going to discuss in detail. First one is container. A container for a pharmaceutical article is intended to contain a drug substances or drug product with which it is or may be in direct contact. Container must be selected with care and after consideration of article's nature and the likely effects of transportation and storage, even for short period of time. A container should be designed so that the contents may be removed in a manner suitable for the intended use of the article in it. It should also provide an adequate degree of protection, minimize the loss of constituents and should not interact physically or chemically with the contents in a way that will alter their quality to an extent beyond the limits given the individual monograph or present a risk of toxicity. The choice of any container for any article is also governed by the likely period of storage of the article during which its quality will not be compromised to a degree where it will be unfit for use. Specifications for the container to be used for any article have not been given, but in certain cases the type of container that is recommended is stated in terms that have the following meanings. Some of the useful definitions mentioning nature of the pack are specified by the pharmacopoeias. First definition is well-closed container. A well-closed container protects the contents from extraneous solids and liquids and from loss of the article under normal conditions of handling, shipment, storage and distribution. Another definition is airtight container. A container that is impermeable to solids, liquids and gases under ordinary condition of handling, storage and transport. If the container is intended to be opened on more than once, it must be so designed that it remains airtight after reclosure. Another definition is hermetically sealed container. A container that is impervious to air or any other gas under normal conditions of handling, shipment, storage and distribution. For example, sealed glass, ampoules, gas cylinder, etc. Another definition is light resistant container. A container that protects the contents from the effects of actinic light by virtue of the specific properties of the material of which it is made, including any coating applied to it. Another definition is multi-dose container. A container that holds a quantity of the preparation suitable for 2 or more doses. Another definition, sealed container. A container closed by fusion of the material of the container. What is single dose container? A container that holds a quantity of the preparation intended for total of partial use as a single administration. Another definition, tamper evident container. A container fitted with a device or mechanism that reveals irreversibly whether the container has been opened. Another definition, tightly closed container. A tightly closed container protects the contents from contamination by extraneous liquids, solids or vapors, from loss or deterioration of the article from effervescence, deliquescence or evaporation under normal conditions of handling, shipment, storage and distributions. The materials that are used in the manufacturing of containers include glass, plastic, metal, etc. Now we are going to discuss individually. First is glass container. It has been the container of choice for pharmaceutical doses form as it has high barrier qualities. It still forms a very desirable and cost effective option. Glass containers may be colorless or colored. Glass is composed principally of the silicon dioxide tetrahedron modified physicochemically by such oxides as those of sodium, potassium, calcium, magnesium, aluminum, boron and iron. Now what are the advantages possessed by the glass container? It is chemically inert, impart no odor and taste to the product, it is non-corrosive in nature, it has a strong and rigid, it is impermeable, transparent and sparkle, no change on aging, it is FDA approved. In spite of certain advantages, it possesses certain disadvantages. It is fragile and high freight cost due to its weight, less pressure safety and impact resistance. Now what are various types of glasses? As per pharmacopoeia, glasses are of 4 types. First one is type 1, that is highly resistant borosilicate glass that is used for parenteral administration. Second one is treated soda lime glass that is also used for parenteral administration. Type 3, that is soda lime glass that is used for dry powders, oleaginous solutions. Next one is 4th, that is type 4 glass that is also known as non-parenteral purpose glass. It is general purpose glass and it is used for oral and topical. A number of glass containers used in pharmaceutical industry includes ampoules, bottles, vials, syringe and cartridges. Another container, second one, that is plastic container. Plastics may be defined as any group of substances of natural or synthetic origins, consisting chiefly of polymers of high molecular weight that can be molded into a shape or form by heat and pressure. Additives present in plastics are plasticizers, fillers, anti-static agents, antioxidants and other ingredients added for special purposes. Plastic containers for pharmaceutical products are made from plastics based on the following polymers. Polyethylene, that may be lower high density, polypropylene, polyvinyl chloride, polystyrene and to a lesser extent polyethylene terephthalate. Now what are the advantages possessed by the plastic? There is a variety of size and shapes with the plastics. They are light in weight as compared to glass and having low freight cost. They are flexible, excellent mechanical strength, non-breakable in nature, having poor heat conductor, essentially chemically inert, strong, rigid, safe in use, high quality and various designs are available. Extremely resistant to breakage. But it also has certain limitations. The limitation is vapor permeation, mostly heat sensitive, drug plastic interaction owing to presence of various additives. Now what are various types of plastics? There are two types of plastic. First one is thermosetting type. Upon heating, they may become flexible but they do not become liquid. What are the examples? Urea formaldehyde, phenol formaldehyde, melamine formaldehyde, epoxy resins, polyurethanes. Another type of plastic, that is second one is thermoplastic type. Upon heating, they may become softened to viscous fluid which gets hardened again on cooling. Examples are polyethylene, polyvinyl chloride, polystyrene, polypropylene, nylon, polyethylene terephthalate, polyvinylidene chloride, polycarbonate, acrylonitrile, butadiene, styrene. Now another type of container is metal container. Metals are also used for the construction of containers. The commonly employed metals are aluminum, tin plated steel, stainless steel, tin and lead. Metal requires the coating and lacquers application for prevention of chemical reaction and corrosion. Now what are the advantages of metal container? They are impermeable to light, moisture and gases. They are strong, rigid, unbreakable, resistant to high and low temperature, light in weight compared to glass containers. Their label printing can be done directly on their surfaces, but it has also have certain limitations. They are expensive, they are having chemical reactivity. Metal containers being used in the pharmaceutical industry includes small elongated collapsible tubes and drums. Now move towards another component, that is closures. A closure for a container for an aqueous parenteral preparation or for a sterile powder is a packaging component which is in direct contact with the drug. It forms a very critical component of the containers. Closure is a device by which containers can be opened and closed. Pharmaceutical closures are essentially elastomeric closures. The selection of elastomer involves consideration of chemical, physical and biological properties with emphasis on the stability profile of drug container system. Primarily, the function of the closure system is to retain the contents safely and prevent hazards resulting in leakage, seepage, spillage, pilferage, loss of quality, purity, etc. by some source of contamination. Closures may be required to be pierced with a needle as in case of multi-dose parenterals. The closures chosen for use with a particular preparation should be such that the components of the preparation in contact with the course are not adsorbed onto the surface of the closure to an extent sufficient to affect the product adversely. The closure should not yield to the product substances in quantities sufficient to affect its stability or to present a risk of toxicity. The closures should be compatible with the preparation for which they are used throughout the shelf life of the product. Now what are the materials that can be used for closure? The closures for containers meant for storage of pharmaceutical products are generally made from the following basic materials. It is cork, glass, plastic, metal, rubber. Now rubber, that is most widely used material as closure. It is made up of materials obtained by vulcanization, that is cross-linking of elastomers with appropriate additives. The elastomers are produced from natural or synthetic substances by polymerization, polyaddition or polycondensation. The nature of the principal components and of the various additives such as vulcanizers, accelerators, stabilizing agents, pigments, etc. depending upon the properties required for the finished closure. There are four main types of rubber which is used in pharmaceuticals. First one is butyl rubber, second one is nitrile rubber, third one is chloroprene rubber and fourth one is silicone rubbers. Closures are available in a 5 basic designs. First design is screw on closure, second design is clamp on closure, third one is press on closure, fourth one is roll on closure and last one is friction closures. Many variations of these basic types exist, that includes tamper proof or tamper resistant, second one is child resistant and third one is dispenser applicator. Temper resistant to the tempering is required for some types of products. Now, what are these child resistant container? Child resistant packaging or the CR packaging has special closures designing to reduce the risk of children ingesting dangerous items tamper evident. Third one is dispenser applicators. A wide variety of convenience dispensing features can be built into closures. Spray bottles and cans with aerosol spray have special closure requirements. Now, some liners are placed with the closures. So, what are these liners? A liner is inserted into the closure to make a seal between the closure and the container on which it fits. It consists of two basic components. First one is backing components, second one is facing components. Closing liners may be of two types. They may be homogeneous liners, they may be heterogeneous liners. And third component is cartons and corrugated boxes. Cartons and secondary pack made up of thick paper in which the filled bottles, vials, ointment tubes are placed, as these are made up of paper and relevant information can be printed over it. It provides strength and opaqueness to primary package. Leaflets containing detailed information regarding the product can also be inserted into it. It is usually of two types. First one is tucked in and second one is sealed. Cartons are finally placed in a bigger container known as corrugated boxes for final shipment, which is sealed with a gum tape. The strength of a corrugated box depends upon the GSM of the paper used and the number of ply used to make it. Type of specifications and methods of evaluation. One ultimate purpose of a specification is an agreement document between purchaser and supplier. A provisional or outline specification is, however, essential. Even when initial development samples are being surveyed, since it provides both a disciplined approach to the examination of materials and components and a record of exactly what was received, used, etc. Component specification. The main specification requirements are the components drawing, artwork, the quality control test and standards. First one is component drawing. Now, the best method for preparing a component drawing is to use a computer system. This will enable rapid preparation and updating of drawing. Several rules should be observed. First one is the following detail should be stated. The detail should be explicit title, specific reference code and version number, date from which drawing is to become impactive, component specification, reference number. Another one is material of construction, terminology used, that is the description of each point of measurement, dimensional limits and units. Second one is a circulation list for each copy of drawing should be available. Third one is enlarge areas of the component drawing to clarify dimensional details when necessary. Fourth one is index each dimension with a number on the drawing to prevent mistaking them for dimensional data. Last one is complete drawing must be checked to ensure that all the details are correct. Second specification is artwork. There must be no errors on the completed artwork. The artwork is the prime reference document for the pharmaceutical company, quality assurance staff and the supplier. Several rules should be adhered. First one is a specific reference code and version number should be assigned on the artwork. Whenever artwork is modified, the version number must be changed. Second one is a circulation list. It should be available for each copy of the artwork. Third one is the date at which the artwork is to become effective is required. Next one is suppliers must not be allowed to prepare their own artwork from the master supplied by the pharmaceutical company. This can lead to artwork errors. The artwork should be computer generated. Another one is the artwork should be color separated. Next one is stick on lettering on the artwork should be avoided. Another one, the exact position of the artwork text, color bands and figures should be shown in relation to the component. Another one, that is print size and type to be used need careful consideration. Type and color of ink to be used must be stated, and if any kind of changes are made to the artwork that renders the old version unsuitable for use. Now move towards another specification, that is third one, quality control testing and standards. If a serious complaint or a product recall occurs, the batch system will help determine the problem source and identify the suspect material on the market. Basic testing is same for both types of components, but additional testing for primary testing such as component compatibility and chemical testing. Both must be tested during manufacture, that is in process control, to ensure the best quality of the product. The critical parameters are, first one is setting the standards, several distinct area. First one is appearance. Appearance, it may be critical, that is unacceptable at any levels. Second one, major, acceptable at low levels. Third one is minor, acceptable at higher levels. Second one is dimensions, that may be critical or non-critical. Now third one, that is measuring components. It is not possible to accurately measure components without trained staff, and variety of measuring equipment such as micrometers, calipers and optical projector. Prior to purchasing equipment, make sure that equipment is reliable, easily calibrated and is of known precision and accuracy throughout its measuring range. That's why the fourth one is precision and accuracy. Once a measuring technique is clearly defined, next aspect to consider is the equipment precision accuracy. Next one is measuring standards. Type of measuring equipment and calibrated, it is necessary to decide the number of components to be measured and the standards to be applied. Another one is sixth one point, that is computerization of measuring equipment. There are several advantages to computerization: prevention of operator transcription errors, rapid recording of all results, audible indication when results are outside limits if required, and instant computer statistical calculation and printout of results. Printout in a standard book format, inclusion of measuring instructions. Next one is component specification layout. There is a tremendous amount of information to include in the specifications. First one is general specification, which states the appearance standards, labeling requirements, packaging and transportation details, and lab testing standards and techniques, as well as any other items that are not likely to change very often. Second one is the artwork should be a separate document with a reference number included on the artwork. Third one, the drawing for a component should also be a separate document with a drawing reference number. Fourth one, the testing methods for both chemical and dimensional checks will be general documents relevant possibly to a whole class of components, example carton, vials and labels. Now how we evaluate these packaging material? The tests generally carried out for evaluation of packaging materials include physical and mechanical testing, weather and aging testing, chemical testing, drug product stability testing. Now we are going to discuss in detail how these are evaluated. First one is glass containers. Generally, hydrolytic resistance and chemical resistance tests are carried out on glass containers. Powdered glass test, water attack test and arsenic test are performed on these containers. Now we discuss individually. First one is powdered glass test. To perform this test, first a specimen has to be prepared. Now how to prepare a specimen? For preparing of specimen, minimum 6 or more containers are to be taken for carried out this test. The exact number of sample is based on nominal capacity of each container and number of packets procured. Initially, containers are rinsed and dried, followed by crushing into fragments. Approximately 100 gram of coarsely crushed glass material is to be taken and divided into 3 equal parts. One portion is to be taken into a pestle mortar and crushed into fine powders. This powder is passed through sieve number 20 and 40. The powder material that is retained on sieve number 40 is to be collected and nearly 10 gram of powder material is to be taken into a closed container. This material is placed in a desiccator until used. Then the sample is spreaded on a glazed paper and magnet is used to remove iron particles from it. Material is sifted to a 250 ml conical flask and washed with 30 ml of acetone. This washing is to be done for 6 times and acetone is decanted. Contents are dried at 140 degrees centigrade for 20 minutes. Dried material is transferred to a weighing bottle. This final specimen is to be used for powdered glass test. 3 samples are to be prepared similarly. This 10 gram of specimen is taken and transferred to conical flask containing 50 ml of purified water. Then all the flasks are to be kept and autoclaved at 121 degrees centigrade for 30 minutes. Further, after autoclaving, the pressure is released and allowed to cool. The flasks are cooled in running water. Now water is decanted from each flask and that residual sample is washed 4 times with 15 ml of highly purified water. Decanted washings are added to the main portion. Then the sample is titrated with 0.02 normal H2SO4 and methyl red solution as an indicator. The volume is required for neutralization and compared with a standard value. Now we will talk about another test, that is water attack test. To perform this test, intact containers are preferred. A minimum 3 or more containers are taken and rinsed with purified water. Each container is filled up to 90% of their overflow capacity. The similar procedure is adopted as for powder glass, except autoclaving is done for 60 minutes instead of 30 minutes. The content of each container is pooled, and from the pooled contents 100 ml of the solution is collected and titrated with 0.02 normal H2SO4 and methylate solution as an indicator. As in case of powder glass test, we have discussed earlier, the volume is required for neutralization and matched with standard value. Now we'll move towards another container, that is plastic container. Numerous tests are performed on plastic containers that include leakage test, water vapor permeability test, collapsibility test, physicochemical test, protection, compatibility test and safety test. Starting with leakage test, 10 containers are selected and filled with water, then closures were fitted and inverted at room temperature for 24 hours. Within this time period, there should be no sign of leakage should be there for any container. Another test, that is water vapor permeability test. This test is mainly done for plastic containers used in blood and blood components. Empty container is filled with the same mixture of anticoagulant solution and sodium chloride injection. Container is closed and weighed, and this container is stored at 5 degrees centigrade with a relative humidity of 50% for 21 days. And lastly, loss in weight is determined and it should not be more than 1%. Another test is collapsibility test. This test is basically performed on those containers which need to be squeezed for product withdrawal. Squeezing it must be 90% of its nominal content at desired flow rate at ambient temperature. Next test is physicochemical test. In this test, the plastic materials are extracted and the extracts are evaluated. The medium for extraction, if not specified in monograph, purified water is preferred as a material and maintained at 70 degrees centigrade during extraction. A specific surface area of plastic specimen is taken for test, and after extraction, following checks are carried out. First one is non-volatile residue. It determines organic or inorganic residue soluble in extraction media and it should not more than 15 mg. Next test is residue on ignition. It is performed when non-volatile residue is more than 15 mg. Third one is heavy metals. It checks the presence of metals like lead, tin, zinc, etc. Another parameter is buffering capacity. It is used to determine alkalinity or acidity of the extract. Another important test, that is protection. When container is anticipated to offer light protection or light resistant container, it must follow the need of light transmission test specified in monograph. Integrity of container, it can be checked by dye penetration test, gas detection test and microbial ingress test. Another very important test, that is compatibility test. It can be determined by regular monitoring of product using chromatographic techniques like LCMS, GCMS. Other changes like change in pH, precipitation, discoloration that may cause drug degradation, it should also be evaluated. Very important parameter. Next one is safety. Biological tests for the containers used for highly risky preparation like inhalation, parenteral and ophthalmic, it is carried out. It is done to check the harmful or undesirable effects of packaging material on products. Extracts are preferred for this test, that is injected or installed in animals. Animals' biological response to plastic material is determined. Next, evaluation is on metal container. For metal container, usually collapsibility test is done. Now, what are the other parameter evaluation for closures? Closures tested on the basis of following parameters: physicochemical test, total extractable determination, it is done by the gravimetric analysis. Next one is stability, it can be checked by visual inspection. Third one is reducing agents. Here purified water extract and potassium permanganate is used. For presence of heavy metals, water extracts are taken. The absorbance, extract absorbance is taken, and identification is done by the use of FTIR, UV spectrophotometry, TLC, etc. Other tests are also performed are Karl-Fischer moisture quantification, S content determination, specific gravity determination. Apart from this, some functional tests are also performed, that is fragmentation, penetrability, self-sealing ability. Again, like container, closure are also tested for safety parameter, and it is done for biological test on extract. Before moving towards next module, that is fourth one, is stability aspect of packaging, I would like to suggest you please visit our website, that is www.cec.nic.in for further reading and learning and more topics associated with your B.Pharm syllabus. Here you will find many lectures, e-contents, LORs and many more, and hope this site will be catalyst of learning for you. Stability aspects of packaging. As far as stability of packaging is concerned, we are more interested in formal stability test at the final stage of any product packing clearance. A stability study should be conducted on the dosage form packaged in the container closure system proposed for marketing, including as appropriate any secondary packaging and container label. It is an ultimate proof of suitability of the container closure system and the packaging processes established by full shelf life stability studies. Stability samples used for evaluation programs must be subjected to quality control inspection. It includes at the program initiation, at diverse stages during extensive storage period, on removal from test conditions for assessment, during opening, after utilization or product application. Development stability testing is used to understand and explore stability in selected pack option. Second one is predict long-term stability. Third one is confirm product protection or need for more protective packs. Example, need for inclusion of desiccants for moisture protection, higher barrier blister films, or need for foil or foil blisters, protective overwrap. Confirm compatibility, identify and explore pack product interaction. Now, what should be the stability protocol? The protocol should include description of packaging, analytical procedures, acceptance criteria and sampling procedures, storage conditions, duration of long-term and accelerating testing, stability commitment and some specific test. In-use test and shipping, photosensitivity, freeze-thaw cycles. And the ICH guideline must be followed that provide reference to stress conditions involving temperature that may be low and high, humidities that is also may be low and high, freeze-thaw cycles, etc. Packs are more prone to change when exposed to a variety of challenges like temperature and relative humidity changes, vibration and compression, the influence of air, light. Changes arising from such challenges may be reflected in the drug product stability. Packaging equipment. To know about the equipment employed for packaging, we have to know first where packaging is involved. Packaging processes involves in various sections like filling section, sealing section, labeling and end of line section. We start with first one, that is filling section. What are the machines that are used in filling section? Machines used to fill bottles or a pouch depending on the type of product. There are certain general types of filling machines. There are auger or agitator filling machines, flow filling machines, tablet fillers. Another section, that is sealing section. Usually after the filling section these machines are used. They are used to securely close and or fasten the product. Some types of sealing equipments that are discussed below are food packaging machines, vacuum packaging machines, blister packaging machines, skin pack packaging machines. Come to third one, that is labeling section. Now, what is labeling? It is an written, electronic or graphic communications on the packaging, and the types of labeling machines that are used are flat glass labeling machine, round bottle labeling machine, side of the glass labeling machine. And now move to a last section, that is end of the line section. It is the final packaging done on individual package and made products ready for the shipment. The types of machine that are preferred are case or box sealers, sleeve wrappers and bundlers, case erectors and box forming machines, case packers, pelletizing equipment. So now friends, it's time to summarize our talk. Pharmaceutical packaging is a challenging and diverse area of packaging design, development and engineering. Packaging affects the quality, stability and identification of drug product. The commonly used packaging materials are container, closures, carton or outer and box. The containers may be made of glass, plastic, metal or paper. The material for closure may include cork, glass, plastics, metal or rubber. There are various tests for determination of quality, integrity and compatibility of packaging materials. The specification and requirement of quality testing depends on type of pharmaceutical materials used. Glass containers are tested by many methods, of which commonly used tests are powdered glass test and water attack test. Plastic containers are tested for physicochemical characteristics, collapsibility testing, biological testing, etc. Closures materials are tested by transparency test, penetrability, fragmentation test, self-seal ability test, extractive test, etc. The requirement of packaging material testing is set according to specification of regulatory agency like WHO, GMP, US FDA and ICH guidelines. Various equipments are used for packaging processes that involve in various sections like filling section, sealing section, labeling and final stage or end of the line section. Thank you friends.